Assalamu alaikum guys. I hope so you all will be doing good. So today we are going to talk about the most important topic, that is about the biological and pharmaceutical importance of enzyme. So let's go and talk about the pharmaceutical and biological importance of enzyme, as it is by me, Rasul Rashid. So first of all, we will discuss about the biological importance of enzyme. So the very first point about biological importance of enzyme is signal transduction. We have some enzymes that participate in signal transduction in various parts of our body. So enzymes are competent in participating in signal transduction, as I have said. So where a chemical or physical signal is transmitted through a cell as a series of molecular it means that the chemical or physical message is sent from cell to cell as a series of molecular event, and ultimately we get the response of the respective enzyme. So the most common enzyme in signal transduction are the protein kinase that catalyze for protein phosphorylation. You may know better about that. So protein kinase help in catalyzation of protein phosphorylation. Another point about the enzyme that is included in biological importance, that is macromolecule degradation. As some peoples, you may say, are lazy to grind their own meal, so the food, or you may say the meal taken into the stomach, or in a big particles, or you may say in a big molecule, so they cannot be broken down easily without the help of enzymes. So large molecules can be broken down by enzyme into small fragments, which can then be absorbed by human body easily. So when these large molecules are broken down by enzyme into small fragments, so the ultimately we can have the small particles which can be easily absorbed and digested. So many nutritional ingredients are in the form of large molecules. Somehow some molecules are also in the large molecules form, so they are degraded into small one by the action of enzymes. So these included protein and sugar and fat. These are macromolecules, you may say, and the macromolecule present in our meal are also broken down by these by some enzyme respective to their function. So we can not be taken easily by human body. So these it means that sugar and protein and fat, this cannot be taken easily if they are in the form of macromolecule. Now I have said that hence these ingredients are broken down by enzymes into smaller pieces before the absorption. So what the process is called, that is catabolism. That this process is called catabolism, in which the macromolecule are broken down into small pieces. Now following the absorption, the small molecules will be used as building blocks to refresh the body through tissue repairing, regeneration and growth, and this process is called anabolism. So the catabolism is followed by anabolism. Many enzymes need to be employed in catabolism, anabolism, such as amylases and proteases. Catabolism and anabolisms are related to each other, so they are happening one after another. So as it is continued, energy generation, the 3 number point, what you said about energy generation, that how enzymes are important in energy generation. So let's see that how enzymes can generate energy for living organisms. Yeah, it's true. How? Let's see. That is about adenosine triphosphate, usually known as ATP, is the main storage form of chemical energy. ATP is a charged battery that can release energy and power the biological activities. And this is clear for us that ATP is a generating energy molecule for us in our body for various functions of our body. Enzymes are the to turn energy into proper chemical forms and store it in ATP molecule. We have certain enzyme that act as a transformer, transformer acting electricity. So enzymes act as a transformer to turn the energy into proper chemical form, because our body, or you may say our body's enzyme, do not want our energy to be wasted, so they are uh, converted into proper chemical form and store it in ATP molecule. Most of these enzymes are called as ATP synthesis. So these enzymes are called as ATP synthesis, mean that they form the ATP molecules. Now another important point about the biological importance of enzyme, that is ions pump. What does it mean? Let's see. Some enzymes in the cell membrane act as ion pumps to move ions across a plasma membrane against their concentration gradient involved in active transport mechanism. So it's an important function about the enzymes, that certain enzymes are acting as ion pumps to move the ions across the plasma membrane. So it is a really important function about the enzyme, so we cannot ignore it, that how enzyme act as ion pumps. So that's another detail if we go to the question how ion pumps are acting as enzyme. So that's a different mechanism and active transport mechanism. So that is a short explanation regarding ion pumps for enzymes. So these primary ion transporter could convert energy from various sources including ATP, sunlight, and other redox reaction to potential energy stored in electrochemical gradient. So these are the pumps that are acting as ion transporter. So they convert the energy like from various sources like ATP, sunlight, and other redox reaction, and these are converted into potential energy stored in electrochemical gradient. As it is still continued, so let's see defense and clearance. Now another important point about defense and clearance, that how enzymes are acting as defense and clearance. So in organisms there are always some non-nutritive substances from external environment are produced by the body itself. We cannot ignore this, that we have some non-nutritive substances from external environment that are engulfed, or you may say ingested into our body, or sometimes our body ourselves produce some non-nutritive or substances which are not needed by our body, so which cannot be either transformed into cellular constituents or employed as energy source. So these are as wasteful, or so we cannot transform into another form, so nor it can be employed as a energy source. So there is a you may say non-nutritive substance, that is heroin. So they need to be converted into product to be excluded or utilized as physiological, pathological, and toxicological component. It's a pretty much simple. Enzyme involving in this procedure might be a single enzyme. So the enzymes involving in this procedure might be a single one, or you may say in combination encompassing various enzymes or enzyme system, or you may say a whole system may be involved, which transform a series of biochemical reaction through oxidation, reduction, hydrolysis, and so on, some other process which can be used by these enzymes to convert the non-nutritive substances to another component as to be utilized for physiological, pathological, and toxicological components. The very important point, and you may all know about the enzyme, that is about speed up chemical reaction, that enzymes are always used as speeding up of chemical reaction. So enzymes are biological molecules, typically proteins, that significantly speed up the rate of virtually all the chemical reaction. Oh, that is pretty much simple for you, that they virtually increase all the chemical reaction, so that take place within the cell. So that must be taken within the cell. So they are vital for life and serve a wide range of important functions in the body, such as aiding in digestion, as I have discussed about the macromolecule degradation and metabolism. As it is still continued, other function. We know that enzymes functions are a lot of, so we can search for our knowledge. So I have written a short for them, that is other functions. So other functions are: enzymes are able to generate movement with myosin hydrolyzing ATP to generate muscle contraction. Muscle contraction can be also brought about by enzyme and transport intracellular substances around the cell as a part of cytoskeleton. Enzymes are important player in many other functions, including immune responses. Immune responses are somehow also taken by enzyme, and the aging process. Aging process are also involving enzymes. Luciferase is the major reason for the glowing of fair flies. So we can also say that luciferase is an enzyme that is used by forensic science for the identification of the victim in major is a major reason for them, and they are only, you may say, only found in fireflies. That is, in Urdu we call it jugnu. So enzymes is a virus. Enzymes in virus are involved in infecting cell or the release of virus particle from the host cell. It's a simple one. Now we are going to discuss about the pharmaceutical importance of enzyme. You may be waiting for this, that what are the pharmaceutical importance of enzymes. So let's discuss about it now. The very first point, that is synthesis of 6-amino penicillinic acid by penicillin A cyclase. It's an important function regarding pharmaceutical. So they are used in industries. Let's see what is that. That is penicillin A cyclase are the group of enzymes that cleave the acyl chain of penicillin to yield 6-amino penicillinic acid. You may pronounce it short form, that 6-APA, and the corresponding organic acid. So this enzyme is used to cleave the acyl chain of penicillin, so which will yield the 6-amino penicillinic acid and the corresponding organic acid. Screening of penicillin. When the penicillin acyclase was screened in microscope producing microorganism, has shown that these enzymes are produced by bacteria. So these enzymes are produced by bacteria, which are called as actinomycetes and yeast and fungi. This compound can be used to create semi-synthetic antibiotics that are effective against the non-penicillase forming bacteria. That's another important point about this. We have a number 2 point about the pharmaceutical importance of enzyme, that is the synthesis of semi-synthetic penicillins by penicillin A cyclase. As penicillin A cyclase have many functions, so another one is that it is used for the synthesis of semi-synthetic penicillin. So semi-synthetic penicillins have been shown to have better properties such as higher stability, easier absorption, and lesser side effects, that are also better candidates against the adaptive microbial resistance to antibiotics. So that's a short description about semi-synthetic penicillins, that what are these semi-synthetic penicillin. So large-scale production of semi-synthetic antibiotics, you may say semi-synthetic penicillin, these are derived from penicillin, are based on the condensation of a beta-lactam nucleus with the appropriate D-amino acids catalyzed by penicillin A cyclase. So we can say that what really penicillin A cyclase do, that is used as a catalyst to form the large-scale production of semi-synthetic antibiotics. That's pretty much simple, and we will not go in detail about the reaction of these various substances to have the semi-synthetic penicillin. So penicillin cyclase is used as a catalyst to form the semi-synthetic penicillin. As it is continued, so enzymes for the production of amino acid. There are various enzymes which are used for the production of amino acids, which are used in industries. Amino acids are the building blocks of life, and thus are indispensable for a proper health and nutrition for both humans and animals. So that's about the amino acid, a short description, or you may say a definition. So these are the 9 essential amino acid, that are L-leucine, L-isoleucine, L-valine, L-valine, L-lysine, L-histidine, L-methionine, L-phenylalanine, L-phenylalanine, L-threonine, and L-tryptophan, that are not synthesized in animals and humans, so therefore must be ingested with food. So we can say we must synthesize artificially these amino acid, or sometime, or you may say many times, we have this kind of amino acid in our food too. So sometimes we have to make these amino acids. Some like tryptophan is made in industries for the in the infant powders to have a food who which infants are not able to breastfeed. So enzymes and whole cell biocatalysts have been important in production of proteinogenic amino acid in pharmaceutical industries. So we can say that pharmaceutical industries are preparing proteinogenic amino acid. Enzymes production for proteinogenic amino acid. That's a little description about now in another point about the proteinogenic amino acid production. So enzymes have been exploited from the last 40 years for the production of L-amino acid in Japan. The production of L-methionine, which is used in special diets, has been carried out using enzymatic resolution with the acyclase of Aspergillus oryza. So it's a short description about the proteinogenic amino acid. As it is continued, so enzyme synthesis for L-leucine. That's another important point in pharmaceutical industry about the enzymes. So the successful co-expression means that they are co-expressed. So Exigobacterium seborrheica leucine dehydrogenase, that's a compound, or you may say a substance, with a bacillus megaterium glucose dehydrogenase in Escherichia coli, E. coli, for the production of L-leucine. L-leucine has a description just below. So this compound is produced by the co-expression of these two compounds. So L-leucine is a basic chiral building block of many pharmaceutically active compounds such as atazanavir, bisoprevir, and talaprevir, so which are potent protease inhibitor for human immunodeficiency virus HIV protease. So these are potent for protease inhibitor for the immunodeficiency virus, that is HIV protease. So hepatitis C, it can be used for also for this hepatitis C virus genotype 1 protease, and hepatitis C NH3 4A serine protease, and respectively. So these can be used about these certain diseases to have inhibition about them. So that was about my video today, and thanks for watching, and keep subscribing. That's a heart from my side. Thank you. See you in my next video. Allah Hafiz.